This is Ryan Gimble, an MD-PhD trainee from Jeremy Rich's lab. I'll be giving an overview of our project on behalf of the co-authors entitled Sorting Nexin 10 Sustains Platelet-Derived Growth Factor Receptor Signaling in Glioblastoma Stem Cells via Endosomal Protein Sorting. In brief, we performed integrative genomic analyses and identified SNX10 as a glioblastoma stem cell, or GSC, specific target. SNX10 functions through post-transcriptional control of PDGFR-beta through endosomal protein sorting and enhances endosomal PDGFR-beta signaling, enhancing GSC survival, self-renewal, and proliferation. Glioblastoma is a highly lethal and incurable brain cancer. Our study is significant as we utilize an integrative multiomics and functional genomics approach as a pipeline to nominate cancer targets. We identify a glioblastoma stem cell specific molecular target with therapeutic utility, both as a direct target and as a possible biomarker for multi-targeted kinase inhibitors. Finally, we discover that endosomal protein sorting controls levels of receptor tyrosine kinases, revealing endosomes as key platforms for oncogenic signaling. Glioblastoma is the most common and most aggressive primary intrinsic brain tumor. It has an extremely poor prognosis, and all therapies are only palliative in nature. One of the factors that contributes to this poor prognosis is the presence of glioblastoma stem cells, or GSCs, within the tumor. These cells promote invasion, angiogenesis, chemo and radioresistance, and support recurrence following therapy, so it's important to target these cells to improve patient outcomes. To understand the molecular processes that distinguish glioblastoma stem cells, or GSCs, from non-neoplastic neural stem cells, or NSCs, we investigated RNA sequencing and H3K27 acetyl chip sequencing data from a panel of 38 GSCs and 5 NSCs. In this situation, H3K27 acetyl chip sequencing marks active enhancer and promoter regions throughout the genome. In panel A, 477 genes were significantly upregulated in GSCs by RNA sequencing. In panel B, around 2,000 gained H3K27 acetyl peaks were identified in GSCs, mapping to 728 genes. Panel C shows overlap of these genes that were upregulated at both the transcriptional and epigenetic levels in GSCs compared to NSCs. These were prioritized for further analysis. To further study this 180 gene overlap, we designed a CRISPR-Cas9 loss-of-function screening library, which was delivered via lentivirus to 6 different patient-derived GSCs. Guide RNA dropout was used to identify genes essential for cell proliferation and viability. We overlapped screening hits across the 6 patient-derived GSCs and focused on genes that were hits in 5 out of 6 of the GSCs tested, indicated in red in panel C, and finally selected SNX10 as a promising target. SNX10 is preferentially expressed in GSCs at the mRNA level, as indicated in panel A, and at the protein level, as indicated in panel B. In GSCs, SNX10 is essential for cell proliferation, as targeting of SNX10 with shRNAs impairs GSC growth. On the other hand, NSCs are much less impacted by the loss of SNX10 expression. We use limiting dilution assays to assess self-renewal capacity, which is a critical feature of GSC stemness. We observe that SNX10 knockdown impairs self-renewal in GSCs, with a less steep line in the knockdown groups indicating reduced self-renewal capacity. To further explore the mechanisms of action of SNX10 in GSCs, we interrogated the Cancer Therapeutics Response Platform, which has small molecule screening data across numerous cancer cell lines. As shown in panel A, in brain cancer cell lines, low SNX10 mRNA expression was correlated with increased sensitivity to several multi-targeted kinase inhibitors, including pazopanib, tivozanib, and lenvatinib, which target receptor tyrosine kinases, including PDGFR. In panel B, SNX10 knockdown diminished protein levels of PDGFR-beta in our patient-derived GSCs, while other growth factor receptors like EGFR, FGFR, and HER2 were not consistently affected. Additionally, we investigated STAT3 phosphorylation as a marker of endosomal-specific PDGFR-beta signaling in panel C. SNX10 knockdown reduced STAT3 activity as indicated by a reduction in STAT3 phosphorylation, suggesting that SNX10 is important for endosomal PDGFR signaling. We studied the cellular localization of SNX10 in GSCs using immunofluorescence imaging and found that it co-localized with PDGFR-beta and was located within early endosomes as marked by EEA1. Panel C shows immunoprecipitation results from PDGFR-beta pull-down experiments, demonstrating a physical interaction between SNX10 and PDGFR mediated by the Fox homology domain of SNX10. We next evaluated how SNX10 affects PDGFR-beta levels. SNX10 knockdown did not impact PDGFR mRNA levels, indicating a post-transcriptional mechanism. We performed PDGFR-beta protein stability assays by treating cells with cyclohexamide to arrest new protein synthesis. Knockdown of SNX10 reduced PDGFR-beta protein stability in panel A and quantified in panel B, suggesting that PDGFR-beta may be more rapidly degraded in the absence of SNX10. Thus, SNX10 controls PDGFR-beta protein stability. In panel C, overexpression of PDGFR-beta rescued cell proliferation deficits caused by SNX10 knockdown. This suggests that PDGFR-beta is the main downstream effector of SNX10 activity in GSCs. We further investigated the effects of SNX10 knockdown on cellular sensitivity to two clinically utilized multi-targeted kinase inhibitors. We found that SNX10 knockdown reduced the sensitivity of these two agents against GSCs. Finally, we tested the potential clinical applications of SNX10. In intracranial xenograft models, SNX10 knockdown extended survival of mice bearing GSC-derived brain tumors. Additionally, in the Cancer Genome Atlas and the Chinese Glioma Genome Atlas datasets, high mRNA expression of SNX10 was associated with poor patient prognosis. These data suggest that SNX10 may be a useful clinical target in glioblastoma. In this report, we performed integrative analyses of RNA sequencing, H3K27 acetyl chip sequencing, and CRISPR sequencing data, and identified SNX10 as a GSC-specific target. We found that SNX10 functions through post-transcriptional control of platelet-derived growth factor receptor beta through endosomal protein sorting and enhances endosomal PDGFR-beta signaling, enhancing GSC survival, self-renewal, and proliferation. For complete details, please refer to the published paper online now at JCI Insight.